Okay, I think we'll go ahead and start. So it's my pleasure to introduce our seminar speaker today, Jason King. Jason just finished his PhD at the University of Texas at Austin in the biomedical engineering department. Jason was mentored by Dr. Tunnell, and while I was there, I was also one of Jason's co-mentors. Jason graduated from, excuse me, in biomedical engineering from the University of Utah in 2017, and then he enrolled at UT Austin. And Jason's work, at least a portion of it, was directed to shaping laser beams for biopsy and ablation of tissues. So, Jason, go ahead.
Thank you for that introduction, and thank you everyone for coming to listen to the talk today. I'm excited to present some of our work on, as Tom mentioned, on laser beam shaping for applications in surgery and for microbiopsy. So start with some background. As you likely know, lasers have some advantages for use in surgical applications, including the ability to precisely remove the tissue, to provide coagulation around the cut, and they can be delivered through optical fibers to reach difficult to reach places, such as in different body cavities. And because of these advantages, lasers have been implemented for laser surgery in many different medical specialties, including ophthalmology, dermatology, dentistry, urology, and cardiology. Today, we're going to be talking specifically about infrared laser ablation. And these lasers in the infrared region usually target water as the primary chromophore. So here we have a plot of the absorption of water versus wavelength, and then the red lines highlight infrared lasers that are used for surgery. And the wavelength or the laser you pick will depend largely on the penetration depth that you're looking for. So penetration depth is inversely related to the absorption coefficient, as the absorption is strongly dominant over scattering at these wavelengths. For these experiments that I'll describe today, we used a holmium YAG laser. This is at a wavelength of 2.1 microns, giving a relatively deep penetration of about 300 microns in soft tissue. And this laser is used commonly in urology because it's very versatile. It can break apart kidney stones of many different compositions, as well as be used for soft tissue surgeries, such as in the prostate or for removing bladder tumors. So when you're optimizing a laser ablation treatment, one important parameter to consider is the ablation efficiency, or the amount of material that you remove per unit energy that you put in. And why this is important is because the energy that you put into the body is going to result in heating. So this infrared ablation is a thermal process where the water within the tissue absorbs the laser, creates a lot of heat, which rapidly vaporizes the tissue and causes this ablation. And so, for example, in lithotripsy, when you're breaking apart kidney stones, water will be absorbed by the laser will be absorbed by water in the area surrounding the stone, and that can propagate outward and damage structures such as the ureter. And in soft tissue, some of your laser energy will be absorbed by tissue around the cut, and this can build up over time and cause significant thermal damage. So there's a need to improve the ablation efficiency in these laser surgeries in order to increase the safety and the speed of these procedures. So one way to do this is using beam shaping. So today we're going to talk both about temporal beam shaping and spatial beam shaping. So first, in temporal shaping, a typical laser pulse has a shape kind of like this one on the top, where it has kind of this shark fin shape where it rises rapidly and then decays over time. And the pulse duration can depend on the laser type. Recently, holmium lasers have started to implement this pulse modulation where you split this single laser pulse into two separate subpulses that are right back to back, and these have specific advantages. Additionally, you might want to have a different laser shape, such as a pulse that builds up slowly with most of the power at the tail end. For spatial beam shaping, depending on application, you may use a typical Gaussian beam, or you may want a flat top shape, or even something like a Bessel beam or an annular shape. So in the presentation today, I'm going to talk about kind of three areas where we applied beam shaping. First is we looked at temporal beam shaping in ablation of kidney stones, or it's termed pulse modulated lithotripsy. Second, we'll go over spatial beam shaping for increasing ablation efficiency. And then finally, talking about spatial beam shaping for a microbiopsy application that we developed. So first, we'll get into the lithotripsy. So just a quick background. Kidney stones happen when there's a crystallization of minerals in the urine. And these can be found in the kidney, as well as in the ureters or the bladder. They can be made of different types of minerals, including calcium, struvite, uric acid, or cysteine. And this is fairly common. Kidney stones will affect about 1 in every 11 Americans sometime in their lifetime. If the stones are small, less than 5 millimeters, they're likely to pass on their own. But if they're larger than about 5 to 7 millimeters, they'll usually require some kind of intervention. So the treatment options can include shockwave lithotripsy, which is done from outside of the body, or shockwaves are focused to the stone and are used to break them up. Or you could do a uteroscopy, where a scope is placed up through the ureter, and using a basket or a laser, you can break apart and remove the stone fragments. If these methods aren't able to remove the stone, then you may have to do percutaneous surgery or even open surgery. So compared to these other techniques, laser lithotripsy has several advantages, including higher success rate and fewer complications compared to shockwave lithotripsy. And it's also able to be effective for all stone compositions and sizes, where shockwave is limited to certain compositions, certain stone sizes, and certain stone locations. And it's minimally invasive as compared to percutaneous or open surgery. So different laser companies have implemented this pulse modulation into their system. So I showed a couple of images here of kind of some marketing images from two companies. So first from Luminous, it shows this image here. Let's see. Here, where the pulse is broken up into two parts. So they say the initial part of the pulse creates this vapor bubble in the water, and then the second of the pulse is emitted through the vapor bubble, so it can reach further and deliver more energy to the stone target. The other advantage that's been proposed is that using this pulse modulation, you can reduce the stone retropulsion, so the stone stays closer to the fiber tip. Shown here, they show it as a virtual basket, so it's almost like there's something holding the stone closer to the fiber. So we had three research questions we wanted to look at in evaluating this pulse modulated laser lithotripsy. First, we wanted to better understand what was the dominant mechanism in this pulse modulated lithotripsy. So there's been two main mechanisms that have been proposed for laser lithotripsy. First is photothermal, which I showed on the on the left here. In this case, what's shown here, here's the optical fiber and the stone surface here, and this is all done in air. So the only interaction we have here is the laser light directly hitting the stone, heating it up, causing this explosive vaporization, which removes the material. And the other mechanism could be a photomechanical or photoacoustic mechanism, where here the stone and the fiber are placed in a water bath. And so when you fire the first pulse, you get this vapor bubble, and the vapor bubble collapses and creates these pressures, which could potentially increase the amount of material removed. The second question we had was, under what circumstances can pulse modulation increase the ablation efficiency? So as I showed in the images, one of the proposed mechanisms is that when the fiber is at a distance from the stone, a first pulse can create this vapor bubble, which increases the transmission of the second pulse. However, some experiments by other groups have shown that this pulse modulation also increases the ablation efficiency when the fiber is in contact with the stone. So we wanted to explore that further. And our third question is, are these phantoms called bigostone a suitable model for laser lithotripsy? So bigostone is a dental plaster that's been used as a phantom for kidney stones in these experiments. It was originally developed as a phantom for shockwave lithotripsy because it has similar mechanical properties to human kidney stones. And it's later been adopted for use in laser lithotripsy experiments. However, the optical properties have not been well characterized. So we wanted to validate if these are actually a good model for laser lithotripsy. So our experiments to answer these three questions involved this setup shown here. So what we had was a stepper motor, which directed the optical fiber originally away from the stone. And then after a series of pulses, it rotates downward. It delivers a single pulse at the stone and then rotates back away. We did this with 1 joule pulses per location using, again, a holmium laser from Luminous. And we cleaved the fiber between each repetition to make sure the amount of energy delivered was consistent. And we compared 3 different types of pulses. First, here's labeled non-Moses, or a typical, just traditional 1 joule pulse. In the middle is a ½ joule pulse, again, just a normal pulse, and we repeated that 2 times per location for a total of 1 joule. And then on the right here is what they call a Moses distance. This is the pulse modulated pulse, where we have this first pulse and then immediately followed by the second pulse. If you look at the time scales here, these are microsecond pulses. The traditional pulse here is about 70 microseconds full width half max. And then we looked at the amount of transmission for each of these pulse modes. So we measured the pulse profile through air and through water. So for this traditional pulse, you can see that through a 1 millimeter water case, this first part of the pulse, which began here, is absorbed by the water. And then after some time, the vapor bubble forms and it's able to transmit. Where here for the Moses distance, part of the first pulse is absorbed by the water, where the second pulse is transmitted. And we found slightly higher transmission for the Moses distance pulse, 84% versus 81% for the traditional pulse. We also looked at the pressure transients generated from each of these pulse modes. And here we saw a drastic difference of about 6 times the amount of pressure was generated from the non-Moses or traditional pulse versus the Moses distance pulse. We then completed these experiments delivering the 1 joule pulse per location under 3 different conditions: a dry stone in air. So in this case, the only interaction is between the laser energy and the stone. A hydrated or wet stone in air. So here we also have water within the stone pores, which can contribute to the ablation. And then third was a hydrated stone in water. So here we also have the bubble dynamics coming into play. And we did this for 4 different stone compositions. We did the stone phantoms, the calcium, struvite, and uric acid stones. After ablation, we used OCT to image the ablation craters, and using a surface detection algorithm, we're able to calculate the volume of each crater. So here are craters for 4 different types of stones. And you can see the size of them varies quite a bit when you're comparing the dense material of Vigo stone and calm versus the more mechanically weak material of struvite and uric acid. So here's a summary of our results. There's kind of a lot of data here, but I just wanted to point out a few key points, some trends that we observed. So first, looking at the Vigo stone or the phantoms, you see as we went from dry stones to wet stones, we got a decrease in the amount of material removed or the ablation efficiency. And when we put the stones in water, we got a further decrease in the ablation efficiency. Where if you can look at the human stones, when we went from dry stones in air to wet stones in air, we actually saw an increase in the ablation efficiency. So the water within the stone is actually aiding in the ablation. And then when comparing pulse modes for the phantom stones, comparing the Moses distance mode in the blue to the traditional pulse in the orange, we saw much larger ablation efficiencies for the pulse modulation for all the cases, even in the case without the water. So there's something in addition to the bubble dynamics that are leading to an increase in ablation efficiency here. But we did not see that effect in the human stones. In the human stones, we only saw larger ablation volumes for the cases where the stone was in water. So our conclusions to these research questions: first, our results support the evidence that ablation efficiency or ablation is primarily photothermal, because our ablation volumes correlated strongly with the amount of energy delivered to the stone and not to the pressure that was generated from the pulse. Second, under what conditions does pulse modulation increase the ablation efficiency? In the native human stones, it only increased it in an in-water environment, which would translate to a case where the fiber is at a distance from the stone with water in between. Where for the Vigo stones, we saw an increase in ablation efficiency for all environments, which would be both when the fiber is at a distance as well as when it was in contact with the stone. And third, how does ablation efficiency vary with stone composition? We observed that Vigo stones differed from native stones, And so in future experiments, we'll need to validate our results from the Beagle stones using native stones as well. Okay, so we're gonna change gears here and get into the spatial beam shaping experiments. So again, in traditional infrared laser ablation, we have this photothermal process where the laser is absorbed by the tissue, creates the rapid vaporization of the water. And this creates this basically an explosion where these small fragments of tissue are expelled. So to increase the amount of material that is removed per pulse, we decided to try using an annular shaped beam. So our idea here is, rather than just getting these particles that are expelled from the region of laser irradiation, we're almost going to be scooping out pieces of tissue. So we cut with this annular shape, and we get this piece of tissue from the center of the annulus to come out, even though it has not been directly irradiated by the laser. So I have a slow motion video here. This is recorded at 50,000 frames per second. This is porcine skin here. And we ablated with the annular beam somewhere here, and you'll see this large fragment of tissue that that comes from the tissue surface. So to do this, we used the optics shown here. So again, we're using this holmium YAG laser at 2.1 microns. We deliver it through a 230 micron fiber, and then connected it to these free space optics here. So we use a lens to collimate the beam, and then 2 axicons to shape it into an annular shape, and then a final lens to focus it onto the tissue. We did these experiments on Beagle stone phantoms as a hard tissue phantom, and we used porcine kidney for soft tissue. For both, we created a flat surface for ablation, and then again used a single 1 joule pulse per location, and compared an annular shape versus a traditional circle shape from the fiber. So comparing the beam profiles for the two, here we have a circular, and here's the annular shape that we were able to generate. And when we did ablation experiments, so here for the hard tissue, for the circular, we see this typical crater shape. It's very rounded. For the annular shape, the craters we see are very flat on the bottom. So what we see here is the laser is actually cutting here on the edges, and we're getting this part in the middle to be ejected as well. For the soft tissue, for the circular beam, we get these narrower and deeper craters, and for the annular beam, we get these wider and a little bit flatter craters. And then looking at the volumes of material removed is very drastic. So for the Beagle stones, we got 183% increase in material removal using the annular, and for kidney stone, it was a 69% increase. Next, to kind of quantify the crater shape, we looked at the crater flatness, which we defined as the width at half of the depth, or the full width half max, versus the diameter at the surface. So a flatter crater has the same diameter at half as it does at the surface, so it would have a flatness of 1, where a conical crater on the opposite extreme would have a flatness of 1/2. And as we expected, we get these much flatter craters for the annular beam. Next, we decided to try to combine the pulse modulation with the annular beam shaping on the hard tissue phantoms. And we saw that they worked well together. We got a further increase in ablation efficiency here in the dark red using the annular shape with the Moses distance pulse modulation. So the conclusions of this experiment was that annular ablation resulted in significantly increased ablation efficiency in both hard and soft tissues. The gains were higher for the hard tissue versus the soft tissue. And the combination of pulse modulation with the annular beam provided an even further improvement. Once again, we would like to eventually repeat these with human tissues to validate, or human kidney stones to validate these results. Okay, so now I'm going to get into the final section, which is where I've done most of my work, which is using a spatial beam shape for laser microbiopsy. So again, talk a little bit about the background here. So we know that traditional histology workflows have several limitations. These are very well known, including the invasiveness of the biopsy, which can be painful, can cause bleeding, require sutures or anesthesia. Additionally, the time to process the tissue is very long. The typical process takes over a day, or you can do frozen sectioning, which takes over 20 minutes. Additionally, a limited amount of the tissue that you harvest is typically analyzed, and it requires a trained technician to prepare the tissue. And then additionally, there can be different processing artifacts. So we wanted to address a few of these problems through laser microbiopsy. And why this is significant, biopsy is, of course, a very common procedure. And for example, in skin, it's very difficult for a dermatologist to determine by looking at it which lesions need to be biopsied. And because these biopsies are invasive, they'll only do a limited number per visit. Additionally, as we said, they usually remove a large piece of tissue. It could be several millimeters in diameter, and may only look at a few sections, which is a small fraction of that tissue. In surgery, over 80% of cancer cases require some kind of surgery, and excising the full tumor is critical. So because of this, there's been many recent advancements for methods to interoperatively diagnose the tissue. So this can include techniques that use mass spectrometry, such as this mass spec pen, different types of spectroscopy, or microscopy. And tissue sampling methods have also been developed that are very non-invasive, such as tape stripping to remove material from the top of the skin and to do genetic testing on, or the mass spec pen, which uses a droplet of water to extract molecules from the surface of a tissue, and then does the mass spectrometry analysis. On the opposite end, traditional tools harvest tissues that are millimeters in size, and again, it can be invasive and limit the number of samples that can be collected. So we wanted to kind of fill this gap between these molecular sampling methods and these large tissue section methods. So we want to be able to collect pieces of tissue that can be used for different types of diagnosis or analysis, while still being minimally invasive. So these advantages of our device come from the size and the scale, as well as from being a laser method, which is non-contact, can provide coagulation be very precise and adjustable in the size and the shape, as well as allowing fiber optic delivery, which could allow it to be integrated with different imaging modalities and as well as robotics or other surgical tools. So this is kind of an overview of the approach. Again, we shape the beam into this annular shape. And when we ablate the tissue, we pop out this small tissue section that's less than 1 millimeter in diameter. You can see the scale here is on the order of a few hundred microns. When it comes out, we collect it directly onto a glass slide, where it could then be analyzed using a variety of methods depending on the application. So it could be conventional H&E staining, could be microscopy, or different types of molecular profiling. So our approach here is the same as in the last section. We use these axicons to shape the beam and focus it onto the tissue. We measured the beam shape we were able to generate at different distances from the optics. So the beam decreases in diameter as it's focused. So to do this, we placed a rectangular capillary filled with a small thin water layer, and we irradiated this with the annular beam and measured the beam shape using an IR camera. So here's our results of that. So on the left here, we have a simulated beam generated in Zemax. And then these are our experimental beam shapes as the beam is focused. So you can see we can get these beams of different diameters. And we have this very nice looking annular shape here. So we place a thin cover slip above the tissue, which we irradiate through. And then when we cut the tissue, our microbiopsy is ejected and it comes out and lands directly on the cover slip. Here we have an OCT image where you can see the scale of this tissue here that we collected. Again, it's on this order of a few hundred microns. And then this is the crater left behind in the tissue. We can, from the OCT images, we can look at the volumes of tissue we remove. And then we use the fast camera to look at the ablation process. Play it one more time. So in our early results, we did some H&E staining of our samples, as well as some fluorescence microscopy. And our samples were very thermally damaged. So you can see here, these are from porcine skin. You can see the epidermis layer and the dermis. However, all the cells are thermally damaged, so you can't see the different cellular features such as the nuclei. And so we knew we needed to come up with a method to solve this, to reduce the thermal damage. And we decided to use cryogen spray cooling, which I'm sure everyone here should be very familiar with. So using this method, we can rapidly remove the heat from the tissue and reduce that thermal damage. So we came with a computational method to evaluate this and to optimize it. So this starts with ray tracing in Zemax, where we put a tissue at the end of our optical train, and we can simulate the scattering and absorption of the light through the tissue. We then put this into MATLAB, where we can look at the fluence versus depth and radius through the beam, and remove the irradiation that's above the fluence threshold or for ablation. And then we have the tissue ejected here to match the volumes of tissue that we were harvesting experimentally. And then we put this into COMSOL, where we can do a computational analysis of the heat transfer and look at the predicted thermal damage. So here's some results from our model. So the first case on the left is without spray cooling. So first we have, this is the temperature predicted in the tissue at the end of the laser pulse. So you can see there's high temperatures generated, which very rapidly damage the tissue. And on the bottom here is a percent damage, where red is 100% damage. When we implement just pre-cooling, again, computationally, you can see that we can limit the heat in this top layer of the tissue. However, over time, the heat spreads, and it still is predicted to cause significant damage. With cooling the tissue after the laser pulse, after the harvest, we can very rapidly remove the heat. However, it's already damaged from this first heating during the laser pulse. And so we really need the combination of the pre and post spray cooling to limit the heat in the tissue and then very rapidly remove the heat that's there. And according to our computational model, with this method, we can preserve a portion of the harvested tissue. We also looked at the effect of scattering. So on the left here is our normal scattering. And then as we reduced the scattering by half and removed it, we're predicted to be able to preserve deeper into the tissue. So this approach could potentially be used with methods to reduce scattering in order to further provide healthy tissue. So here's our implementation. We used fuel injectors and R134A cryogen. Here we have a porcine skin sample, our cover slip, and then our beam shaping optics are in here. So we're able to provide the pre and post cooling and collect the sample onto the cover slip. We then looked at the samples using microscopy. We wanted to do virtual H&E microscopy to provide images that look like H&E. So our method here, we used acridine orange as to stain the nuclei, and SR101 to stain the collagen in the cytoplasm. So this is a very simple process that doesn't require any tissue pre-processing. It requires about 2 minutes to do the staining, and then we image with a confocal microscope and then false color to create these virtual H&E images. So the example I showed here is, this is porcine skin before ablation. I just cut a cross section through the skin and stained it. So A is the acridine orange image, which simulates that of hematoxylin. B is the SR101, which is an eosin analog. And then we combine them and then color them with this pink and purple color of H&E. So here are results of our ablation experiments. So first, this is the unablated skin. This here is an example of the bulk skin after ablation. So here's the crater in the tissue. And we see there's some thermal damage, evidence of thermal damage here in the epidermis on the sides. We didn't see much thermal damage at the base of the tissue. And then C and D are examples of our microbiopsies. So you can see in this one here, we'd be looking at the epidermis from the top. You can see the nuclei are preserved, and we can see some of the same features. And then on the outside, there's kind of this different color from the SR101 channel that indicate thermal damage. And then here's another example that we see in some of our tissues where the tissue kind of folds over itself. So there's some mechanical disruption. You can see different pockets of cells mixed with collagen. And we also did this in porcine kidney. So again, A is our unablated porcine kidney. And then B is a crater after the ablation. So you can see there's some differences in the color along the crater, indicating some thermal damage. And then C and D are examples of microbiopsies we collected. So you can see, again, the nuclei are preserved. There's some thermal damage around the edges. And then with kidney being mechanically weak, we saw a lot more mechanical disruption. So pieces of tissue that look like this, where it's kind of been broken apart. But we were largely able to reduce the thermal damage. So in conclusion, our laser biopsy with virtual H&E allowed observation of histological features. The cryogen spray cooling aided in preserving the tissue integrity. We expect this to be compatible with different types of analysis techniques, such as sequencing. And in our future work, we'll look at collecting from different tissue types, adding different types of analysis, again, such as mass spec or molecular profiling, as well as develop methods to limit the mechanical damage of our harvested tissue, and eventually lead to development of a device that could be used in the clinic. So I'd like to thank my mentors, James Tunnell, Tom Milner, as well as Stepan Parek and Joel Teichman for their guidance in these projects, in addition to the members of the Tunnell and Milner labs. You recognize some familiar faces here in this image, as well as our funding, and to be happy to answer any questions that you have.
Thank you, James. Questions or Jason? Thinking of your supervisor,
Yeah.
James. Questions for Jason?
I have a question, if possible.
Okay, Emo, go ahead.
So, first of all, congratulations. Very good talk, very brilliant work. And for brilliant work, there are many questions. So, first, about biopsy. Laser biopsy. So if I'm not mistaken, you got small samples, about 100 microns inside. Is it correct? So you have shown small dimensions. And what temperature was for inside these particles? If the temperature is high, you may have altered material. Your biopsy will give wrong result. You will have denaturation due to procedure. What's the temperature during laser biopsy?
So to just make sure we understand your question, is it that you're wondering what the temperature is in the tissue during the microbiopsy?
Yes.
In the, I think there were two. There were two tissue types, right?
Yeah. Yeah, so we didn't actually measure experimentally the temperature in the biopsy during the experiments. All we have is the computational results, which definitely have some limitations. There's a lot of approximations we included there. So that was this slide here, kind of the top row, is what the computational results show. So it showed with the cooling, we can keep this top part cool, but that it heats up to 100 degrees here at the bottom. But again, the computational results I wouldn't expect to exactly match what we're seeing experimentally because of the different limitations.
So if you have 100 degrees, you must have denaturation in this hot zone. So it's maybe in nature tissue was different structure. So your histology will show wrong result in this case. That is my question. How can we trust this biopsy?
I'm not sure. I can't hear very well. I'm not sure.
I think maybe go back to your histology that you did after in showing the cells and what kind of thermal denaturation you did or did not observe after the process of the pre.
Yeah. So our goal here in these experiments was to try and preserve the tissue as best we could. So the method we used, because we didn't measure the temperature directly, we decided to use this microscopy as the analysis to determine if we were getting too much damage or not. So again, in our, for example, this image here in C, we only saw evidence of the thermal damage in the microscopy here at the edges, where the middle looks largely preserved. When we didn't use the cryogen cooling, this would just look like one solid color. We wouldn't be able to see any of these details here. And then kind of a next step, we'd like to do some other things like try to look at the cell viability. So separate the cells and do a viability test. Look at the DNA to make sure that it's not denatured so that we can do genetic testing. So those are kind of some next steps to make sure the tissue is preserved to the extent that we need.
Thank you.
Yeah, thank you. Very nice work. I like the idea of optical biopsy, like optical punch biopsy.
Mm-hmm.
I wanted to ask, I didn't see the numbers for the depth. They were like at the intersection between the screens. So how deep does it go?
Yeah, the depth is usually around 200 to 300 microns.
Okay. So comparable with a lateral.
Yeah, so it's comparable to the penetration of the laser. So if we wanted to cut shallower than that, we could use a laser with higher absorption. If we wanted to cut deeper than that, we'd probably have to have multiple pulses to kind of drill down through the tissue.
Yeah, that was the other question. How much you can control, how controllable that is.
The depth right now we can't control too much. I think you would want to control it more with the laser wavelength and the penetration.
Yeah.
Mm-hmm.
I see. Is there a particular target application you have in mind, or you're exploring any?
Yeah, I mean, it kind of depends to what extent we can improve it and what kind of form factor we can put it in. So we'd like to get it into, I mean, right now it's free space optics and they're fairly large. So we'd like to get like something that could go through an optical fiber. So you could go, for example, in the esophagus, you could take small, it's an area where they take random biopsies from many locations. So that would be one. Another kind of ideal case we see is using it for surgery, to both do the surgery to remove the tissue as well as analyze it as you're going. So you could kind of do the surgery, and on the way, you're pulling out these small pieces of tissue that you could look at to determine if they were cancer or not. Like, should I keep cutting? Should I stop? Kind of that real-time feedback.
Yeah. Okay, we'll talk more like half an hour. Thank you.
Thank you.
Nice work.
Sean.
Hi, Jason. Thank you for the talk. For the laser surgery, you're using like a microseconds laser. I believe it's going to produce ultrasonic wave. Have you guys ever thought about using this laser-induced photoacoustic wave for monitoring the beam in terms of the timing of the beam and spatial shape of the beam? Because I believe it's going to relate it with the ultrasound signal being generated. So have you guys?
So I just want to make sure I understood. You're asking if we've used ultrasound to monitor the temporal profile of the beam and the spatial as well as spatial?
Yeah.
No, I haven't thought about that.
Because you showed one data is like pressure wave. What is that pressure wave? It's a acoustic wave.
Right.
So I kind of believe that the strength of the acoustic wave is proportional to how much energy being deposited there. Also, the frequency of the ultrasonic wave could relate it with like what's the pulse duration of your laser. So you can actually real time catching this photoacoustic wave to monitoring the beam itself.
Yeah, that would be interesting because there is a lot of mechanical aspects of this harvest process. So we've tested this on weaker tissues like brain, and the pressure waves that are generated are so high that it basically shoots out a stream of tissue. It doesn't hold together. So that would be an interesting, if we knew what kind of pressures we were generating, we could kind of optimize that based on the mechanical properties of the tissues. So I think there's a lot of interesting work that could be done there.
Yeah, sure. Thank you.
They use the photoacoustics to almost get the tissue mechanical properties to synthesize.
Yeah.
Yes.
Sorry. So I was wondering, from your experiments, is this annular beam the best possible beam for cutting, or does a better beam exist out there that you just haven't come across yet? And did you experiment with that?
Yeah, that's a good question. One of our initial thoughts was like, what if we could, when they do histology, they take these 5-micron slices. Like, could we actually just like cut out a slice? And I think because of both the mechanical aspects and the thermal aspects, I don't think that's possible, but definitely, yeah, different shapes would be interesting to take into consideration. I think for us, the annular beam's nice because it creates this even pressure around the edges, and they kind of all push towards the center and push the tissue right out. But yeah, I think the ideal thing would be to take a section of tissue if that was possible.
One thing you might try is you have your vessel beam usually in the annular part in the back aperture of your focusing objective. But if you slide that forward, you actually get curved field vectors. So you might actually have a different pressure profile. So
okay.
I don't know.
So you put the vessel beam closer to the...
Yeah, if you defocus it, make it a bad vessel beam, in other words. You might change the way you ablate things.
Interesting.
Yeah, I have a question. For the volume of the microbiopsy, can you somehow control it to be bigger or be smaller?
That's a good question. Again, with our current system, it's a pretty narrow range that it successfully harvests these biopsies. If you focus it much tighter, you start to basically ablate the whole volume, like you don't get anything. And if you get it much larger, there's not enough energy to push the tissue out. We're kind of limited by that. But I think with different, again, I would think it would be better to change the wavelength and also change the laser type. So if you wanted to get a smaller biopsy, you'd want to go maybe to a fiber laser. I think you could be able to focus it tighter than we can with our with ours, because we have to go through a 230 micron fiber, and then to focus that and still have enough working distance, it's harder to get it much tighter than what we have.
Have you tried to ablate after the first pulse? Let's say you have the creature, then you shoot another pulse. Will you be able to pull another piece of tissue out?
I've tried that a little bit. I haven't been successful, but that's not something we've done a lot of work with yet.
Okay.
Yeah, you'd want to be kind of moving your optics down as you did it. So like right now, if you shot twice in the same spot, your second fold would be, the tissue surface would be too the optics and the tissue after every fold.
Sure. If you pre-stretch the tissue, what do you have an idea what's going to happen? Or even somehow put internal pressure, will that change the volume?
You could, if you put pressure, you would change the scattering properties, and maybe, yeah, I'm not sure about the optical properties might change a little bit. As far as the mechanical effect of it, I don't think it would make much difference, but it's an interesting question.
Thank you.